Hello, I'm Adeline Boire. I'm a research scientist at INRAE, the French National Research Institute for Agriculture, Food and Environment. I'm investigating the structure and assembly of proteins in grains. Today, I will tell you about intrinsic disorder in plant proteins. What we learn in school is that proteins need a highly specific 3D structure to have a biological function. This is the structure-function paradigm. Proteins are formed by amino acids, hydrophilic amino acid, hydrophobic amino acid, positively charged, negatively charged amino acids. Amino acids are bonded covalently to form an amino acid chain. These amino acids interact with each other and form secondary structures such as alpha helix, as shown here, or other structures like beta sheets. Secondary structures are arranged in a 3D manner to form tertiary structure. Here represents three ways of representing a single protein, an enzyme, for example. This type of protein, fully ordered, are also called globular proteins. The 3D structure of a protein will be complementary to other molecules and will drive the function of the protein. For example, a substrate will fit the catalytic site of an enzyme, which will lead to the reaction. However, in the late 90s, we realized that unfolded domain of proteins also play important roles in protein function. The terminal domains of this protein lack of a stable conformation and fluctuate over an ensemble of conformations. This is what we call intrinsically disordered regions or intrinsically disordered proteins. Intrinsically disordered regions are protein domains which are disordered, while intrinsically disordered proteins are fully disordered. There are several hallmarks for intrinsically disordered proteins. First, they have a low content in hydrophobic amino acids. They also display a low complexity sequence and contain high content in polar and charged amino acids. It's now well accepted that globular proteins only account for 30% of total human proteomes. 10% is completely disordered, and a whole range of proteins display an intermediate range of disorder degree. Most experimental tools have been designed to characterize the structure of a protein, like X-ray diffraction. But usually they fail to characterize a structure. Instead, you can predict the disorder of a protein using softwares and the amino acid sequence of the protein. You enter a sequence of the protein into a server, and it will predict the portion of your protein that may be ordered or disordered. Now, if you draw a graph with the y-axis, the order degree, from flexible to rigid, and the x-axis being the hydrophilic degree, from hydrophilic to hydrophobic, you will find globular proteins from milk and egg on the right corner of the graph. They are predicted to be rigid, except for casein. While seed proteins display a range of flexibility. Here you will find cereal proteins, like wheat and barley proteins. Here you will find pea proteins. And finally, rapeseed proteins. Proteins from grains are predicted to be more flexible and more hydrophilic than egg and milk proteins. The scientific questions that we want to tackle through our research are the following. Are plant proteins disordered or partly disordered? What is the role of this disorder in their biological function? And what are the implications for food processes? Several people are contributing to this research project: PhD students, research scientists, engineers, and technicians. For more information on intrinsic disorder in proteins, have a look at the list of reference provided below this video.